Okay, so now let's talk a little bit about molecular activity and what a turnover number is. Okay, turnover, we're going to talk about what's called a kcat, which is a turnover number. So this refers to the number of substrate molecules converted to a product in a given unit of time on a single enzyme molecule when the enzyme is saturated with substrate. So the number of molecules of substrate converted to a product each second per mole of enzyme, per molecule of enzyme. So in other words, how much? So the enzyme can take a certain amount of this substance and convert it into something else. Okay, so with regards to enzymes, when you think, if you see the word or the term kcat, it's really referring to how much. How much can that enzyme carry of substrate to product, okay? So let's use an example of this equation down here, okay? So we have right here, here's our enzyme, the Ca. This is what we're going to actually learn a little bit about this once we get to cardiovascular physiology, respiratory physiology, and acid-base balance. This is the main equation for that, okay? So this right here is called carbonic anhydrase, okay? So this is the enzyme we're talking about here, 2 different locations in the body. So carbonic anhydrase catalyzes this reaction, so it speeds it up, right? So if you didn't have this enzyme, it would occur, but very slowly. And if anyone knows, is anyone, you can pontificate for a second, what is this reaction, right? I just mentioned it has something to do with acid-base balance. So this reaction is actually a major player with regards to what I just said, acid-base balance and maintaining proper pH levels. So we'll talk about it a little bit more during pulmonary physiology, but I'm going to keep going with it for a second. So as you exercise, as you exercise intensely, you produce a lot of excess hydrogen ions as well as lactate, right? So excess hydrogen ions decreases our pH, okay? We become very acidic. Okay, and somehow we need to get rid of those hydrogen ions, okay? So, but let's just look at it here for really quick for a sec. So we have, we're going to go this way. We're going to start over here. So we have all these hydrogen ions that are building up in the blood, okay? So what are we going to do? We're going to combine it here with a bicarbonate ion, and then we're going to use carbonic anhydrase to combine hydrogen with the bicarbonate ion to produce carbonic acid right here. This is carbonic acid, okay? And then that's how it likes to travel in the blood. So carbonic acid, how you like to travel, hydrogen likes to travel in the blood is in this form called carbonic acid right here, okay? So then as it travels in the blood and it gets to the level of the lungs, we're going to use carbonic anhydrase that is stored within the lungs, again, to then break it apart into carbon dioxide and water, okay? All right, we're going to use, the carbonic anhydrase to split it up into water and CO2, okay? And that's what we expel, right? So when you take an exhale, you breathe out, what do? Every breath you take, you expel some sort of water as well as carbon dioxide. So this is really how we're helping maintain acid. This is one major way how we're maintaining acid-base balance is exhaling CO2, and this is how it travels in the blood so how are we getting rid of a lot of that excess hydrogen is because here, this is our buffer, okay? So when we complete metabolic testing, for example, let's put it into real life forms here. So when we complete metabolic testing in the lab, the amount of carbon dioxide we are producing is really actually representing how much hydrogen ions we're producing within the blood with exercise. So if I were to hook you up to the metabolic cart, and again, hopefully we'll be able to do it this semester, what you'll be able to do is you'll be able to see as exercise intensity increases, you're going to see an enhanced respiration rate, and we can actually look at different numbers. And one is how much carbon dioxide is being produced each second, right? So that actually gives us an indirect number of how much hydrogen ions someone is actually producing based off of the intensity of the exercise, and that will give us an idea of what energy systems are dominant at what point in time. Okay, so the number we look at on the metabolic cart is something called an RER, which is the respiratory exchange ratio. It is the amount of carbon dioxide, it's the ratio that represents the amount of carbon dioxide produced to the amount of oxygen that's consumed. Okay, we'll go over that again later in the semester. I'm just using this as an example right now and just kind of giving you a couple couple little metabolism tidbits here, right? So we'll see the higher an RER is, that means that the person who is exercising or is using mainly anaerobic pathways, okay? And the lower the RER is from a scale from 0.7 to about 1.1, it's closer it is to a 0.7, they're mainly burning fats as a fuel, all right? And we're gonna, like I said, get into that later on. But that's an example of a reaction that's important within our body that relates to this class, okay? All right, now we're going to talk a little bit about training and enzymes. So let's use some examples. Enzymes within our mitochondria. So let's talk about aerobic training and the Krebs cycle, okay? So when we aerobically train, do we produce more enzymes, or does each unit of enzyme do more work? So with chronic training, do we produce more enzymes? Can we produce more enzymes biologically, or does each unit of enzyme do more work? So meaning, does each enzyme that we already have become then more efficient? Okay. Actually, the answer is, in most cases, it's sometimes one or the other, or it can also be a little bit of both. So we are biologically capable of producing more enzymes, as well as our enzymes becoming more efficient. So meaning they can do more work, bringing more substrate to product, or we produce more so we can have a quicker rate, okay? All right, so you can get more units of enzymes created, or training the athlete may cause each enzyme to take more substrate to product. So for example, the enzyme is becoming more efficient, okay? So right down here is a lovely photo of the Krebs cycle, sometimes called the TCA cycle, Krebs cycle, all that lovely stuff. Here, you don't need to know every single turn of this. Once we get into aerobic metabolism, which we will be getting into, I will explain what you need to know about Krebs. You don't need to know each enzyme. You need to know, probably a key player or so, and, why is it a key player, but you don't need to know all of these substrates and all of the enzymes, how they're regulated and whatnot. That was something lovely I did in graduate school and had to know. You do not need to know that. So I'm just going to explain to you what you need to know and why is the Krebs cycle important with regards to metabolism. That's what I care about. Okay, so the next thing, some basic definitions. If you see this out there, or when you read a research article, you'll be able to understand what they're talking about with regards to enzyme kinetics. Oxidation versus reduction. This probably rings a bell back from chemistry and whatnot and some basic biology. Oxidation, we are removing an electron. Reduction, we're adding an electron. Remember, an electron is a negatively charged particle. So by removing an electron, we have actually done something positive. And actually by adding an electron, we have actually added something negative. I know it's kind of seems a little backwards there, but electron is a negatively charged particle. So when you add one, the reaction is becoming you're adding something negative. Okay, so there's actually some different ways to classify enzymes. We can classify them systemically by number. We'll go over the basics of what you need to know. For example, we'll go over the practical uses for these enzymes. And if we were in an exercise-specific biochemistry class, we may go in more depth, but here's what we really care about is practical use. Okay, so what we're going to do is we're going to head into start video number 2. We're going to please head on over to that, and we're going to start with enzyme cofactors.